Nuclear chemistry continued, medical and scientific applications of radioactivity. In 1895, Wilhelm Röntgen, and there are two ways to spell this with the umlaut shown here, those are the two dots over the O, or as an OE, but it's pronounced Röntgen. Wilhelm Röntgen discovered X-rays in 1895. So this is a year before Becquerel discovered radioactivity as a general concept of being particles from the nucleus of atoms. Now remember, we now know that that X-ray radiation is actually from the electron cloud. You won't be required to know the distinction, but it's a good idea to understand that this was not considered the discovery of radioactivity when X-rays were first found. But X-rays, we now know, can be used to image dense tissue, such as bones and teeth. We have more sophisticated methods that can to image a number of other tissues in our body as well. But the very first medical image that was taken in the year that Röntgen discovered it, 1895, was of the hand of this woman, Anna Bertha Ludwig. And so you can see the difference between a modern X-ray shown at the top and the very first medical X-ray ever taken of Ms. Ludwig's hand. As it turns out, Ms. Ludwig wasn't Miss Ludwig, she was actually Mrs. Röntgen. She was Wilhelm Röntgen's wife, and she was good enough to place her hand under here. And when she saw the X-ray, being German, she said, "Ich habe meine Tod gesehen," which means, I have seen my death, because it was the first time anyone had imaged the skeleton of a living organism. And this was a big deal, an enormous medical discovery. And in fact, shortly after this, about 15, 20 years later, Marie Curie, the famous Marie Curie, developed a portable X-ray machine that she put in a van, an ambulance, and ran around Europe with this van, X-raying soldiers who were involved in World War I. And she ended up saving many, many lives as a result of having the X-ray available for these soldier patients. Now, modern X-rays have really expanded in technology, and we use them not only for medical purposes, but for scientific and even security applications. So shown here are just some of the many uses of X-ray technology. If we look at the third picture here, medical CT, this is the CAT scan, the computed tomography, which is essentially taking X-ray slices throughout the body, and then a computer combines these slice photos of different slices of your body, different X-ray photos, and combines them into one three-dimensional image of your entire body, or whatever portion is being imaged, so that the physician can then, the physician or the analyst of whatever type, can look at these images and rotate them in space and look in great, great detail at the X-ray images of a person or even an animal in order to help better diagnose and monitor diseases. We also have modified versions of the X-ray used for mammography, and if you look in the far right, we all know that going through airport security, X-rays is one of the common ways that your baggage is examined for possible weapons or other dangerous materials. And then on the far left here, we have what's called X-ray crystallography. And this is a technique where crystals of a certain chemical are first prepared. So crystals are grown of a single substance, and that could be a very small molecule or something as large as a protein. And you make these crystals, and they're in a very specific lattice form where the molecules are all in a very repeating pattern. And then you hit this with a beam of X-rays, and the X-rays, being very high ionizing radiation, hit the crystal, and they result in enormous amounts of ionization of the molecule, resulting in electrons and fragments of the molecule being dispersed in all directions. And then a detector that surrounds this material can determine what the blast pattern is of the particles from the original molecules, and from that blast pattern actually calculate back what the original structure might be. And so this would be as if you were to take a shotgun, shoot it in an apple, and have a person come into the room who has never seen an apple, look at the apple splayed on the walls all around the room and say, hey, what did this thing used to look like? And with computers, that computer can say, well, it used to be this shape, the shape of an apple, and essentially X-ray crystallography is doing the same sort of thing, only far more sophisticated, where very complex molecule structures have been determined by use of this technology. So use of radionuclides in science and medicine is now very important, and one of the reasons why it's so useful is that we have very sophisticated ways of measuring radioactivity, very sensitive ways to measure even small amounts of radioactivity. And ionizing radiation, which we've already talked about, breaks molecular bonds, in some cases can cause changes in the cellular or biochemical processes in a cell that result either in damage to the cell or killing the cell, and ultimately can result in cancer, can either result in producing cancer, or if it's used as a treatment, can be used to kill cancer if used properly. So ionizing radiation can do either a lot of damage in a good way or a lot of damage in a bad way, and we also have very good ways of measuring it. So combining these two concepts, we can use the information from radionuclides to develop new technologies. One of the most significant damage to molecules, of course, is DNA, and that's why the cancer comes. So we have these different uses. We have scientific uses of radioactivity, and we have medical uses, and some of these overlap. One of the earliest scientific uses of radionuclides is what we call radiometric dating. Radiometric dating is used to date ancient materials to determine when they were first produced, by looking at how much radioactivity of a certain isotope is left in the material since it was first produced. And if you look at different isotopes, you can look back and answer the question, when was this first made? C14 is used if you want to go back as far as 50,000 years. But beyond that, it's not very useful. So we have to use other isotopes like uranium-234, potassium-40, uranium-235, uranium-238, and these allow us to date rocks or wood or human artifacts or fossils to tremendous ages, even in the billions of years. We also have radiation therapy. So while radiation can cause cancer, we can target it to just the cancer tissue and kill the cancer cells with minimal damage to the healthy tissue that might be surrounding it. And we have a number of isotopes that are typically used in treating cancer in radiation therapy. Cobalt-60, because it can produce gamma radiation, iodine-131, which can be attached to certain molecules, which will, if injected into the body, will migrate towards the thyroid where iodine is sequestered. Cesium-137, gold-198, these all can be used to treat tumors to try to shrink these tumors down. We also have a variety of imaging technologies like MRI, SPECT scans, and PET scans, which are scans that detect certain types of radiation, and gamma cameras, which detect gamma rays, obviously. And by injecting different isotopes that might be attached to other molecules into your body, these molecules will migrate to different tissues in your body, and then you will go into an instrument like an MRI or a SPECT scan or a PET scan or a gamma camera, and these imaging devices will determine where that radioactivity is, and if you attach these to certain molecules, these molecules will migrate to tissues like the brain, the heart, or tumors, which tend to be very fast growing. We also have a variety of techniques under the category of medical diagnostics. So these are essentially blood tests and other tissue tests that can be used to detect the concentration, the presence and concentration of certain hormones, antibodies, enzymes, other proteins, and even small molecules like drugs. And these require the use of radioactive labeled molecules, but these techniques are known as RIA, radioimmune assay, and IRMA, immunoradiometric assay. And these techniques, because they use radioactivity and they're used by technicians in the lab, there's a certain amount of danger with these, and as a result, over the last 20 years, most of these tests have been phased out. There are only a few labs still doing tests with this material, and these have generally been replaced by much less harmful materials that produce, instead of radiation, they produce color or fluorescence or chemoluminescence, which is essentially small amounts of light that can be detected with different imaging devices. And then related to that, we have other types of biochemistry assay methods. The original methods, some of the original methods for determining the sequence of DNA and RNA were in fact done with radioactive materials, especially radioactive phosphorus P32. And these again are also more or less phased out. We now have very sensitive fluorescent methods that are now being used, and these methods are actually extremely automated nowadays, so that instead of taking months and months to do large sequences, we can actually sequence the entire human genome in a day now with some of the most advanced DNA sequencing instruments, which use fluorescence and not radionuclides. So in terms of radiation therapy, there are a number of different types. We have external bombardment, and this includes two of the most famous types of bombardment that both use gamma radiation, the so-called gamma knife and the cyber knife systems. And shown here on the right at the top is a gamma knife, and what happens is you have radioactive cobalt, which is producing gamma rays, and it's in a holder in the material, which is moved around whatever target tissue needs to be irradiated. And the reason why it's moved is you want this beam to always be pointing at your tumor, but remember, the beam will also hit all the other tissue that is along that beam. So if you are always moving the beam direction, but always pointing at the target, the beam will always go through the tumor, but not always go through the same different tissues in your body, especially if you're doing it to the brain. So this way, minimal damage happens to the healthy tissue, and you're only always pointing at the tumor tissue. We also have implants, and implants can take the form of wires or balloon catheters or a number of other devices that are inserted into human beings. Shown on the bottom here are beads that are used to treat prostate cancer. But you can use these different radioactive materials to produce these different shapes that are implanted into people near the tumor in order to irradiate close to the tumor tissue without irradiating too much of the healthy tissue. We also have what's known as palliative care, and palliative care means relief of pain. So this is typically for cancer patients where the patient is still going to be terminal. Unfortunately, they cannot be saved, but you want to do the most you can to help relieve their pain while they're still alive. And certain types of treatment with some of these radionuclides can be used in order to irradiate certain tissues to limit how much pain is actually produced from those tissues to give some kind of relief to your patients. And then we have internal radiation. So these are molecules that might be a hormone. They might be some other molecule that normally would, if you were to inject them, would migrate through your bloodstream to a certain tissue. But if you first chemically attach a radionuclide like I-131, you can attach this to thyroid hormone, for instance, or some precursor molecule similar to thyroid hormone, and this will migrate to the thyroid. And if you have thyroid cancer, this is a good way to kill the thyroid cancer without the radiation being close to other healthy tissues in your body. As I mentioned before, we have different types of scanners, the SPECT scanner and the PET scanner. And SPECT scanner, SPECT stands for single photon, so that's a single gamma particle, emission, computed tomography. So computed tomography basically means a computer-generated three-dimensional image of where the gamma particles are emitted in your body. And what happens is you're injected with a material that releases gamma particles, and that material is a certain molecule that will migrate to the tumor, and then you can image where the tumor is. PET scan is very, very similar, but instead of using a gamma emitter, you use a radioactive nuclide that emits positrons. And now you're not responsible for knowing details of positrons, but these are antimatter versions of electrons. So they're actually positively charged electrons, the opposite of a normal electron. They're the same size, they're the same, they have many of the same characteristics, but they're positively charged. And these can be detected with a PET scanner. So positrons, antimatter is real, and people get injected with this every day, not the same people, but every day in order to detect tumors. And one of the classic ways to do these is you inject these materials like the gamma emitter in the case of the SPECT scan. It emits the high energy photons, and say you're using a radioactive iodine like 123 or 131, which both produce gamma radiation. If you're putting iodine in your body, it will migrate even as plain iodine, as iodide, the ion, will migrate to the thyroid and be turned into thyroid hormone, and that will result in an ability to image the thyroid in your body. In the case of PET scanners, which are producing these anti-electrons, these anti-beta particles, and these are labeled beta plus to distinguish them from a beta particle. One of the most common ways to use positron emitters is to take fluorine-19, a positron emitter, chemically attach it to glucose. And fluorine-19 has a half-life of only 110 minutes. So what happens is this material is actually made with nuclear reactions early in the morning in one part of the country, and then it is transferred immediately after it is synthesized to a chemical lab that will attach it to glucose and purify it. And this is all done within a couple hours. And then it is sealed in a vial, put in an airplane, sent to the hospitals that will use it later that day. And then this is injected into the person all before all of the radioactivity goes away because it has such a short half-life. It's injected into the human being, and because it's glucose with this fluorine on it, the body thinks it's just plain glucose, and it will be sent to the parts of the body that need glucose the most, the parts that burn the most glucose, and those are the brain, the heart, and tumors, which tend to be fast-growing. Unfortunately, it also shows up in your urinary tract system because you excrete excess glucose that might be in your blood. So it's not always useful for certain types of tumors like bladder and kidney because there's going to be so much accumulating there anyway, it's too hard to see anything else. But it is useful for a wide range of other tumors. And shown here is an image of someone that has glucose with fluorine on it. What's called FDG, 19-FDG. This is injected, and what you see is the heart is lit up, which you'd expect. If you look at the top, you see the brain is lit up because they use a lot of glucose. You can see the urinary tract at the bottom is lit up, but what you also see, and this is the bad part for the patient, is that there are other internal organs that are also lit up, and in this case, you see the metastases of the liver, which have now have resulted as a result of the original colorectal cancer in this patient. So you can see this patient has an enormous number of small tumors spread out throughout their abdominal cavity. In addition to fluorine-19, other compounds with carbon-11, another PET emitter, are also used, and some of these are actually used, they're attached to molecules that will migrate to the parts of the brain involved in Alzheimer's. So this is just starting to be used for imaging Alzheimer's as well as a non-invasive method. And with that, we'll stop here.